Alright, hi everyone, it's Marvin here, and I hope you all had a good Halloween. I'll be presenting this week's article, which is titled Comparison of Human Embryonic Stem Cell Derived Cardiomyocytes, Cardiovascular Progenitors, and Bone Marrow Mononuclear Cells for Cardiac Repair. So, just as an overview, I'll be going over myocardial infarction, the three different cellular sources for cardiac repair that the researchers wanted to look at, and as well as the study design of what they wanted to look at and how they structured their experiments and give an overview of the techniques that they used. Then I'll be going over the hypothesis and objective of the study. And lastly, I'll be going over the importance and impact of the study. Moving on, I'll be going over the results, which will be mostly an interpretation and summary of figures 1 through 4. And then moving into a discussion which highlights some of the conclusions and findings from the article, as well as some of the unaddressed questions or limitations, and my own suggested additional experiment. And I'll wrap it all up by talking about some future directions for their work and for future studies as well. So just as a background on myocardial infarction, we'll start with the basics. It's more commonly known as a heart attack, and it's due to a blocked flow of oxygen-rich blood to the heart. So this blockage can be due to a buildup of plaque, which is mostly made up of fat, cholesterol, and other substances in the arteries that actually feed the heart. The effects from a heart attack can be seen pretty quickly, as the damage is usually seen within 30 minutes of the blockage. So for diagnosis, there's a few tests that can be performed. There's an electrocardiogram, which is an ECG that looks at the heartbeat of the person. There's blood tests that can be performed, as well as chest x-rays for visualization. There's angiograms as well, which is where they inject these dyes into the coronary arteries. And after taking an image, you should be able to see whether or not there's like blockages in these arteries. And it'll basically show up as kind of a cutoff of the flow of blood, which could be indicative of a heart attack. And there's also some less used tests, such as an exercise stress test or a cardiac CT or MRI. Now, there's also some treatments that can be performed. There's medications to improve blood flow and as well as dissolve clotting. There's coronary angioplasty, where they use a balloon catheter and inflate it inside of the artery in order to push the plaque to the walls of the artery and better open it up so that blood flow can be restored. And the same thing goes for stents, where a stent is placed over the balloon so that the balloon can inflate inside of it and place the stent. And then from there, the stent can provide a better structure for the artery, as well as open it up for improved blood flow. There's also some surgeries to treat arteries, and as well as some recommended lifestyle changes. And from my own research, I also saw that myocardial infarctions can be classified into different types, and as well as there's different signs for heart attacks as well. So if you want to check out this link and do your own research as well, that might be kind of beneficial to better understand the disease state. So moving on, there's of course cellular treatments that are being developed as well, which is highly interesting to look at. There's 3 different cellular sources that could be used and that were identified in this article, as well as many others. There's these human embryonic stem cell cardiomyocytes, there's these cardiovascular progenitors, and there's human bone marrow mononuclear cells. So now I'll be going into detail of these 3 different cellular sources and talk about their upsides and downsides. So for these cardiomyocytes from ES cells, there's a lot of research that's actually been done about them. And several studies, including this one that's been cited in the article, have demonstrated some potential features of them, such as remuscularizing the myocardium, engrafting, providing better contractile function, having very quick effects, and as well as forming these electromechanical junctions. And as you can see from some of these figures, they've actually demonstrated these results with, one-to-one coupling, some vascularization into the graft where the stem cells are located, and as well as some remuscularization here. However, they do have some downsides. There's a failure to improve already diminished cardiac function. So as you can see, between these different cell groups here that are used, there's not really any significance. And each of these graphs represent, a heart function, or some kind of indication for heart function. So as you can see, there's not really any significant differences between the groups. And here as well, even with these graphs where they have different measurements of the heart or different performance metrics, each of the groups kind of perform similarly and don't have any significant, p-values. So next up are the cardiovascular progenitors derived from ES cells. So the nice thing about these is that they have a restricted capacity to differentiate into only these 3 different kinds of cells. So they can only differentiate into cardiomyocytes, smooth muscle cells, and endothelial cells. And they also have the potential to remuscularize an injured heart and promote revascularization of an injured area of the heart. So looking at these 2 features could be very promising for developing therapies and treatments. And lastly, we have this sort of gold standard human bone marrow mononuclear cell. So there are some key features that make these cells very attractive for use. They're easily accessible and have a great safety profile. And in preclinical studies, they've demonstrated efficacy, which is really important. But for clinical trials, they're also the most frequently used cell source. But if you look at the downsides, there are some inconsistent results for improved cardiac function. So this could be a really big downside, especially for the purpose of this paper, and it's also very unclear for the mechanism of action behind these cells. So even though everybody can demonstrate efficacy, there's not a lot of research that's been done to look at the mechanism lying underneath it. And on top of that, there's not a lot of creation for new cardiac muscle with these cells. And that doesn't really help generate a lot of contractile force for the heart, which is very important as well. So next up, I'll be going over the objectives and the hypothesis. So their objective was to compare the fate of these 3 cellular sources, these cardiomyocytes, progenitors, and mononuclear cells. And their hypothesis was that these progenitors can provide better repair since they can contribute multiple components of the myocardium. And another sort of hypothesis that they didn't really indicate, but what I found from the article was that they wanted to show that there's other cell sources besides these gold standard ones that can be used for cardiac repair applications. So since everybody's using these ones, but nobody really understands the mechanism of why they're so beneficial, it might be worthwhile into looking into these other 2 groups, the progenitor cells and the cardiomyocytes. So for the study design, first they took these ES cells and differentiated them into progenitor cells as well as non-cardiac cells. With these non-cardiac cells, they served as a negative control group. And the cardiac progenitor cells could be differentiated into cardiomyocytes. They didn't really look at endothelial cells or smooth muscle cells for their applications of use. And they also looked at this 4th group, which were the mononuclear cells from bone marrow. These served as the positive control group because they're the most commonly used ones right now in any kind of application. So kind of stepping through what their thinking was and how they wanted to proceed, first they needed to characterize the cells that they obtained from their differentiation methods. So we'll talk about it more in the figure 1 recap, but they looked at all these markers, which are kind of highlighted in orange here, to characterize their cells. And this helped confirm that, the cells that they were looking at were actually cardiomyocytes or progenitors or non-cardiac cells or the mononuclear cells. They also wanted to look at cardiac function immediately after cell injection. And they did this with echocardiography as well as some other methods that I'll detail later. And then 1 month after transplantation and injection, they wanted to look at histological analysis and as well as revascularization to just to understand, how these cells, progress throughout their time within the heart. So the importance behind this research is that heart disease is the leading cause of death for men and women. Approximately 735,000 Americans have a heart attack each year. So if they could really nail down, which cellular source is better to use, they could, come up with a treatment and, come up with better optimized protocols that could help, reduce this devastating disease. And there's not really been any studies prior that compared these three cellular sources to each other within the same animal model. Different research groups have only used, one cellular source and used like macaque models or different kinds of animal models as well, but nobody's really compared the three sources together. And the impact is that they wanted to be able to look at host cardiac remodeling as well as cardiac function after transplantation. And being able to compare the three sources is so important because then they could make recommendations and kind of establish, their own kind of like gold standard for use. And in the following videos, I'll be going over the results as well as the discussion.